afternoon, everyone.
Good afternoon.
So, welcome to our highly anticipated webinar, Nanotoxicity, a challenge for future medicine, brought to you by Heredity Biosciences. I'm Sushree Swagatika, your coordinator for today's session. And I'm thrilled to have each of you join us for what promises to be an enlightening discussion. First and foremost, I would like to extend our deepest gratitude to our distinguished speaker, Dr. Piyush Kumar Gupta. Dr. Gupta's expertise in biomaterials development and nanotoxicology is truly invaluable, and we are honored to have him share his insights with us. Thank you, Dr. Gupta, for your unwavering support and dedication to advancing scientific knowledge. I would like to acknowledge our convener, Dr. Gopal Krishna Purohit, the visionary CEO and co-founder of Heredity Biosciences. Dr. Purohit's groundbreaking work in clinical genomics and infertility research has made him a truly inspiration for our field. We are privileged to have him leading our organization and guiding towards greater achievements. Before we move into the discussion, I would like to provide a brief overview of today's schedule. We kindly request all participants to stay engaged until the end of the webinar. Following the conclusion, our session will find a feedback link in the message box. Your input is valuable to us, and we ask that you take a moment to share your thoughts to receive your certificate. Please ensure that your feedback forms are complete in full of the consideration for certificate. As we move into the questionnaire session, I request everyone to maintain respectful and organized dialogues. Allowing for a seamless exchange of ideas, your active participation is crucial for success of our webinar. And we encourage to make the most of this learning opportunity. So without further delay, I would like to hand over the virtual mic to our esteemed director, Dr. Gopal Krishna Purohit. Once again, a warm welcome to all the participants. Let's make this journey of knowledge together. Thank you, and over to you, Dr. Purohit.
Good afternoon, everyone. I hope the clarity of my slide is visible for all.
Yes, sir.
Thank you, Sushree, for the splendid introduction. First and foremost, I extend a heartfelt welcome to all attendees of today's distinctive webinar. I am Dr. Gopal Krishna Purohit, the CEO and co-founder of Heredity Biosciences. It's a pleasure to have each and every one of you join us today. In today's webinar, we investigate into the transformative domain of nanotoxicity, a challenge for future medicine. As nanomaterial gain grief in biomedical applications, undergoing their potential adverse effect is crucial. Addressing these demands in interdisciplinary collaborations and innovative strategies for the safe integration of nanotechnology in healthcare. A sincere thank you to our esteemed guest speaker, Dr. Piyush Kumar Gupta, head of the Center for Development of Biomedical at Sharda University, Greater Noida, India. Your acceptance of our invitations has greatly enriched today's webinar, and the entire Heredity Biosciences team is truly grateful for your presence and expertise. Dr. Piyush, your valuable contributions has significantly elevated the quality of our webinar. Thank you for being in the integral part of this insightful session. Now, the audience, the highlight of webinar session Today's webinar is centered around the intricate study of nanotoxicity, a challenge for future medicines. Explore the potential risk associated with the use of nanomaterials in biomedical applications. As nanotechnology advances, understanding the adverse effect of nanoparticles on human health and the environment becomes crucial. This topic delves into the interdisciplinary efforts required to mitigate this risk and ensure the safe integrations of nanotechnology in healthcare.
So nanotoxicity has revolutionized the scientific landscape, so broadening the inquiry into the various applications across the medical field. Some of the key applications include the drug delivery systems. It is the encapsulated drug for targeted delivery. It concerned to the persist over the potential toxicity over prolonged exposure. Next one is the diagnosis tools like nanoparticle enhance the medical imaging for diagnosis purpose to use to that through this biocompatibility and long-term safety remaining crucial. Next one is therapeutic use. Innovative cancer treatment through targeted drug delivery systems, demanding safety assessment. Next one is implemental device, like nanomaterials integrate into the implemented enhanced biocompatibility, necessitating through the potential nanotoxicity. Next one is regenerative medicines like nanotechnology tissue engineering and regenerations requiring careful assessment of nanomaterials interacting to prevent adverse effect. Next one is environmental remediations like nanoparticles to use for the environmental cleanup, but concern over the ecological impact and persistence necessitate crucial applications. Next one is personalized medicines. It is used for the told diagnosis and therapeutic use to use it and demanding rigorous evaluations for safety and efficacy. Next one is biological sensors, and it is used for the enhancing biosensors, yet careful design to testing the crucial of material potential toxicities. And gene therapeutic use for these nanoparticles like gene delivery to the vectors, but concern immunogenicity and genotoxicity to the comprehensive safety assessment. Last one is biomedical research, like advancement in research, but ethical and responsible use to nanomaterials is vital to the minimize risk. Once again, I heartful thanks to Dr. Piyush Kumar Gupta for enriching our webinar with your presence and expertise. Your time and knowledge are truly appreciated. Now, audience, before we begin our webinar and introduce esteemed guest speaker today, allow me to provide a brief overview of Heredity Biosciences. As you know that Heredity is established in 2018 and rebranded in 2021. Heredity Biosciences is a premier provider for the research and training services for hospitals, life sciences institutes, and pharmaceutical firms. It is a trusted name of medical and scientific community. Our sessions team specialized in genetic, proteomics, and bioinformatics, and we are committed to quality and aimed services and collaboration. Right now, we are collaboration with the Heric Life Sciences Private Limited to impact good innovations and global health, life sciences, and healthcare research. Heredity Biosciences proudly operate from two locations. The main office is Mayfair Lagoon Road, Bhubaneswar, and a newly branch opened for the Guwahati, Assam. It is inaugurated last year. This expansions inside significant milestones reflecting process of our growth and boosting our capabilities and fostering collaboration with different scientific institutions, research institutions, and we anticipate the meaningful contributions to advancing life sciences through this exciting expansions also. Heredity Biosciences excel in life sciences and healthcare. As you know that we are offering comprehensive research and training services like life sciences services as well as healthcare services. Research services, we investigate in the crucial area of clinical microbiology, infection genetics, cancer genetics, fertility genetics, and conventional qPCR-based DNA studies. Everything is available in our websites. Heredity Academy is one of the Odisha's leading institutes providing for the training, comprehensive hands-on training for students and professionals, and specialized modules like clinical embryology, that is male infertility, histopathology and immunohistopathology, and qPCR-based genetic diagnosis, personalized digitations program also available to advance laboratory practice to the students and preparing to the students of a dynamic landscape and biosciences. To join as a Heredity Biosciences and go to the journey that combine academic accuracy with the hands-on applications, empowering you become the future leading to the leader of the field of this life sciences. We invite everyone to visit our central facility and we witness to the following features like world-class laboratory facilities and high connectivity study environment also. Complete research learning facility, well-trained, experienced and trainer and demonstrators also available every time. Well-equipped genomic and microbiology lab, complete hands-on individual experimental exposure, publications assistant and placement assistance that available, and experienced fast and state-of-art infrastructure and designed enhanced to the learning and research experience. In the past 3 to 6 months, we are very initiative to this Heredity Biosciences also witness a successful spread to the placement activities. This is the witness of this postdocs I'm interested here. Over this last 3 months also, numerous students completed their training and secure position in the esteemed establishment organizations, including fertility clinics and pharmaceutical companies. Their achievements stand to the testament to the effectiveness of our training programs. We extend to the heartfelt congratulations all the students and wish them the journey filled to the prosperity and fulfilled to their growing career. May their contribution to the field of biosciences or healthcare to the both impactful, rewarding. So, cheers of the success and promising to the path, career path ahead. So, Heredity Biosciences have initiative for this every times and research and publications. In-house research and publications. So we have more than 30 publications. In this month, we have published in the three publications, new publications. And right now we have a 3 patent already filed and ongoing research already going on for future things. And we are collaborating with the research institute, hospital institutes, so that we will move on the research and research sectors. Now, audience, now we introduce for this guest of speaker. Ladies and gentlemen, it is a great pleasure that I introduce our esteemed guest speakers for today's webinar, Dr. Piyush Kumar Gupta. Dr. Gupta, currently the head of the Center for Development of Biomaterials at Sharda University, Greater Noida, has forged an academic journey that stands as a testament to his unwavering dedication in the field of life sciences. Dr. Gupta's expertise is on biomedicals development and nanotoxicity, where he has made remarkable contributions. His inventive spirit has led to the development of numerous technologies, with 5 granted and total 9 Indian patents published in recent year. Furthermore, he has authored 140 SCI and Scopus indexing pre-reviewed articles and 7 book chapters, solidifying his reputation as a prolific researcher. He's recognized for his exceptional contributions. Dr. Gupta has gave with several international and national awards, including the Best Researcher Award of Sharda University, the INSA Visiting Scientific Award, the RSC Nanoscale Horizon Award, and the Speaker Nature Award, and the Young Scientist Award, among many others, have his dedications. His dedications to advance in the field in evidence through the editorial response as an editorial board member, his trial and fancies, fronters, springers, and elsewhere journals. Additionally, Dr. Gupta served as an active pre-reviews for 40 international journals and hold membership in prestigious scientific societies such as like Royal Society of Chemistry, American Chemical Society, and European Society for Biomedicals. Dr. Gupta's influence extended globally through his more than 50 international and national invited talks, where he shares his wealth of knowledge and insight with the audience worldwide. Today, we have the privilege of benefiting from Dr. Gupta's expertise as a share invaluable insight in a webinar titled Nanotoxicity, a Challenge for Future Medicines. We eagerly anticipate the knowledge we will impact and look forward to stimulate discussions ahead.
Yeah.
This is a team Heredity Biosciences. Thank you.
Yeah, thank you very much. So, Dr. Gopal, can I share my PPT?
Yes, yes, sir.
Yeah. So it is visible, like my PPT is visible for everyone.
Yes, sir.
Yes,
Okay.
sir.
Yeah, thank you very much. So before starting about nanotoxicity, and as Dr. Gopal has given about the brief introduction about my academic profile, I'm very thankful to him and also the Swagatika that they have invited me for giving such an invited talk for Heredity Biosciences, and especially for the UG, PG, and PhD students. So as I am going to discuss about the nanotoxicity. You can see here, like I will discuss about its introduction and what are the effects that we can see on the human health. And the most important part that is required, like how to analyze the nanotoxicity. And as we are using several drugs and also the nanodrug carriers or the tissue engineering products in our medical sectors, so how to analyze or how to study their toxicity? So we need to understand about its toxicokinetics inside the human body. And several computational approaches has been established nowadays to study this toxicity assessment for the nanomaterials. And what are the important tools in nanotoxicity analysis that I will cover in my during my presentation. So before going to the introduction, and we are well aware about the nanotoxicology, that if we are studying about the toxicological effects of different nanomaterials, that can be either the organic nanomaterials or the inorganic nanomaterials. So such type of study comes under the nanotoxicology field. And currently, this is a rapidly expanding area because whenever we discuss about any cosmetic products, or either we are discussing about the medical products, and even any products that we are using for the environmental at environmental levels, we are just trying to develop the nanoparticles. So these nanoparticles are widely utilized in our daily life. If you take from any level or at any sectors, and due to which it is very important concern that we need to understand its toxicity inside the human body. So the toxicity can be understood only on the basis of the multidisciplinary scenario. So before going to the analyzing its toxicity, we need to understand that what types of nanoparticles present in our environment. So as you know that you can see here, there is a natural nanoparticles, there is a incidental nanoparticles, and the engineered nanoparticles. Natural nanoparticles are made by nature, where that can be either inorganic or organic natures. As you can see that lignin nanoparticles, or you know very well aware about cellulose. So cellulose nanoparticles, also the pollen grains. And if we discuss about in the animal kingdom, so there is a chitin that that is also can be formed into the powder form, and that is also the nanoparticles. So natural nanoparticles are not more toxic to the human health, but whenever we are using these nanoparticles for any biomedical applications, we need to understand their degradation property. Because whatever we are using for the biomedical application, we need to know like how the nanoparticles is degrading, and once it is degrading, what are the byproducts it is releasing. So whenever the natural nanoparticles degrade, their byproducts are not toxic in nature. But whenever we discuss about the incidental nanoparticles, so how the incidental...
Sir, sorry for the inconvenience, but the slide is not changing, sir.
Okay. So now you can see the slide?
Yes, sir.
Okay.
Thank you.
Yeah. And can you tell me now it is changing?
Yes,
Yes,
sir.
sir.
Yes.
Okay. So here I was discussing about the natural nanoparticles, incidental nanoparticles, and the engineered nanoparticles. So you can see these natural nanoparticles can be either also or also of organic or inorganic nature. Similarly, the incidental nanoparticles and the engineered nanoparticles. But whenever we discuss about the natural nanoparticles, these are obtained from the nature, and whenever these nanoparticles are degrading, they are not releasing the toxic byproducts. But when we discuss about the incidental nanoparticles, whenever they degrade, they release a lot of toxic byproducts. So for example, if we take the petrol or the diesel, and when we burn the petrol or diesel, it releases the carbon nanoparticles. So you can see that these carbon nanoparticles, when it enters inside the body, it can generate lot of free radicals, which leads to the production of higher ROS level, and it can disturb the cellular homeostasis. So it can be threatful for the human body, and it can generate several catastrophic diseases. So incidental nanoparticles we can obtain from the burning of the fossil fuels, and if we are burning any natural products, so like the wood or the plant product, so then it generates lot of incidental nanoparticles. And engineered nanoparticles we generally make inside the lab. So these nanoparticles can be either organic or inorganic nature. So as you know very well, like a lot of researchers have made different types of metal nanoparticles or metal oxide nanoparticles. For example, you can take the zinc oxide nanoparticles, silver oxide, or you can take the ferrite nanoparticles or the magnetite nanoparticles. All these nanoparticles are engineered nanoparticles because you can prepare these nanoparticles according to your this, like what type of physical chemical properties you want to include in these nanoparticles, or you want to like functionalize these nanoparticles with any type of ligand, for example with any antibody or aptamers. So here we can do any type of engineering on the surface of nanoparticles, or also in the composition and in the size of nanoparticles. So such nanoparticles are called the engineered nanoparticles. So if we can see, the natural nanoparticles are not toxic, and also after degradation they do not release any toxic product. But whenever we take the incidental nanoparticles, and when we just degrade these nanoparticles, it releases lot of toxic products. And engineered nanoparticles can be toxic also, but it depends what type of precursor you are taking to synthesize these nanoparticles. If they are precursor after when you use these precursors and you synthesize the nanoparticles and they are releasing any type of byproducts, it can be either toxic or not toxic in nature, but that we need to study inside the human beings. But before using for the human beings, like to check inside the human beings, we need to understand its toxicity level on the in vitro and the in vivo models. So as I discussed about the different types of nanoparticles, we also study their physicochemical properties. So here I have listed the 4 important physicochemical properties that is affecting the nanomaterial toxicity. And you can see here one of the most important property is composition. Either these nanoparticles are made up of organic compound or either the inorganic compound. So you can see here, like we can utilize the lipid to synthesize the liposome or the solid lipid nanoparticles or the solid lipid nanocarriers. Even lot of researchers have used the polymers to synthesize the polymerosomes or the micelle or even the polymeric nanoparticles. Similarly, you can utilize different types of metals like a gold or the silver or the iron or the zinc or the magnesium or the calcium to synthesize the metal or other metal oxide nanoparticles. So here, as you can see, the liposome or micelle, both are the organic nanoparticles, and while you can see here the silver, iron, and the gold, these all are inorganic nanoparticles. So when these nanoparticles degrade, their byproducts, either it can be toxic or non-toxic, but before utilizing, you need to understand their toxicity. The second important point is size. So as you can see, if the size of the nanoparticles is higher, then once it will enter inside your human body, it can trap inside the alveolar cells or also the windpipe. So when it just trap, then it start recruiting lot of immune cells, and due to which you can get the upper respiratory tract infection also. So the size of the nanoparticles is also very important. And if the size is lesser, it can easily cross your any gap junctions between your cells, and it can reach to the kidney from where it can filter out. But when the size is higher, it cannot easily cross the gap junctions also, and it will entrap inside the kidney cells also, due to which after sometimes you can see it is just affecting your kidney, and also once it reaches to the liver cells also, it also affects the liver. So inhaling lot of nanoparticles that is present in the aerosol, it also create lot of respiratory disorders, and also it can lead to the it can affect to your vital organs. And we can see the next the important part, and the most important physicochemical property, which is surface chemistry and the charge. So charge is also an important point to understand the nanomaterial toxicity. So the charge on the nanoparticles can be either negative, or it can be either positive, or it can be either in neutral form or in the deuterionic form. So mostly whenever we synthesize nanoparticles, we need to understand these nanoparticles we are using for which application. If we are using for these nanoparticles for the intravenous application, we always try to synthesize the negatively charged nanoparticles, because when these nanoparticles will enter inside your blood, and when it will interact with your RBC, then these nanoparticles will repel with the RBC cells, so that it will not create any hemolysis. And if you mistakenly synthesize the positive nanoparticles, then these nanoparticles will interact with the RBC cells, and then it will damage the RBC cells and it will release the hemoglobin. So the surface chemistry and also the surface chemistry lead to the charge on the surface of nanoparticles, and that can be either negative, positive, neutral, or deuterionic. But we need to understand what type of charge we require on the surface of nanoparticles, and that we can select based on the application. So the charge is responsible due to the ligands that we are attaching on the surface of nanoparticles. And also, what are the precursors we have used to synthesize these nanoparticles? It also impart an important role on the surface charge of the nanoparticles. And the other most important criteria is, say, are you making the sphere shape of the nanoparticles, or either you are taking the cubical shape or the polyhedron shape, rod shaped, or either the disc shaped? Because sphere can easily cross to the gap junctions, and it can be filtered out from your body easily through the nephrons. But if you are making any other size, then might be it can entrap inside your in alveolar cells, or either in the windpipe, or either it can just entrap into your liver cells and also the kidney cells. And also, whenever we are synthesizing the nanoparticles, size also we consider. Why? When we are selecting any type of applications, we need to understand which has the more surface area. If you want to upload or encapsulate more drugs, then you try to make spherical nanoparticles so that it can easily deliver the drug to the cells at maximum level. So we need to understand what type of size we require to develop the nanoparticles, and it is based on the application. But if we see the composition, size, charge, and the shape, all these physicochemical parameters are responsible for the nanomaterial toxicity. So the most important concept that we need to understand when we take these nanoparticles, how they enter inside our body, and then how it is interacting at different levels. So you can see these nanoparticles can enter in your body either through the ocular way, or either you can inhale these nanoparticles, or you can take from the mouth, or either the skin, or either you are injecting inside the blood. So there are different types of exposure pathways by which these nanoparticles can enter inside the human body. And whenever these nanoparticles enter inside the human body, first of all, these nanoparticles interact with your organs, and then it reaches to your cells. And once it will just uptaken by the cells, then inside the cells, they interact with different type of biomacromolecules. So like if it is interacting with the protein molecules, it just denature the protein molecules, and it can imbalance the cellular homeostasis. So you can see in the below cartoon, like these nanoparticles, when interact with different types of biomacromolecules, they can lead to the generation of reactive oxygen species. And also you can see these reactive oxygen species either can be found in the singlet oxygen, or either the superoxide radical, or the oxygen radical, or the peroxide ion, or the hydroxyl radical. So it is continuously generate the ROS, and due to which it can disturb your cellular homeostasis, and which can be a important concern to generate different types of cardiovascular, endocrine disorders, and even it can be toxic to your different organ systems. And sometimes we see, like if you are using any metal oxide nanoparticles, and when you take inside the human body, then these nanoparticles can degrade. How it will disintegrate into the different ions, and these ions can slowly can enter inside the cells, and where it will interact with at different cellular organelles. And after interacted with different cellular organelles, it can disturb the physiology of these organelles, like the functions of these organelles, so that it is just disturbing the cellular homeostasis, or we can say the cellular systems. So disintegration is also an important part. And the third is signaling pathways. Like these nanoparticles, when interact with different proteins, and which is present on the cell surface as in the receptor form. So these nanoparticles, when interact with these receptors, they just disturb the downstream cellular signaling pathway, so that the cells undergo the senescence stage, and then cell undergoes the apoptosis, and due to which cell death occur. So there are different types of exposure pathways by which the nanoparticles can enter inside the human body, and it can affect the it can interact with the organ level, and then cellular level, and then biomolecular level. And you can see here there are different types of effects you can see, like a chemical effect, physical effect, and the biological effect, where you can see the ROS generation and the disintegration of nanoparticles, and even it is affecting the cellular signaling pathways. So when we go to the effect of these nanomaterials on human health, you can see the toxicity can be seen either in the form of apoptosis and in the form of necrosis. So as we know very well that apoptosis is a programmed cell death, while necrosis is not a programmed cell death. So generally inside our body, the cells are aging and it is continuously going under the apoptosis conditions. But whenever the nanoparticles enter inside the human body and then they disintegrate, they can either induce the apoptosis and necrosis. So apoptosis cannot be harmful for our body, but necrosis can be very, very harmful for our body. And apoptosis also, if the healthy cells are going in the apoptosis stage, then it is also a serious concern. So what they do when the nanoparticles enter inside the cell, either they will damage the intracellular organelles or either they will lyse the intracellular organelles. And when they enter inside the nucleus, they damage the DNA, and it can lead to the mutations inside the DNA, and due to which the cells undergoes in the proliferation stage, and due to which it may convert into the malignant stage from the benign condition to the malignant stage, and it can be responsible for the cancer disease. So the damage to the nucleic acid and also the plasma membrane and the membrane receptor and the ion channels, all these aspects is responsible for the cellular toxicity. And when the cells are undergoing the toxicity, you can see its toxicity level on the organs and or the organ systems. So when cells are undergoing the toxic effects, then it will damage the different organs like a lung, liver, brain, spleen, kidneys, and the skin. And what effect you can see, like these organelles, these organs are undergoing the inflammatory conditions. And sometimes whenever we are taking lot of nanoparticles, we are inhaling lot of nanoparticles, we get the allergic reactions. And sometimes we also get the anaphylactic shocks. So you can see here, I have given a cartoon, like when nanoparticles enter inside the human body, what type of diseases they can cause. So you can see from the brain diseases, it can create either the neurological disorders, Alzheimer or the Parkinson. And if it is entering inside the lung, you can see the asthma, bronchitis, cancer. And when it is entering inside the circulatory system, then you can see the arteriosclerosis, vasoconstriction, even the high blood pressure and the thrombosis. It can also change the heartbeat, and it can damage the kidneys and the liver. And also you can see the inflammation on the skins, where you can see the inflammation of the dermis and also your blood vessels. And you can see its effect on the gastrointestinal systems, and that can generate also the different types of cancerous diseases like colon cancer. It can also create the blood cancers and the lung cancer. So if we see nowadays in our environment, there are a lot of nanoparticles present because we are using a lot of fossil fuels, and these fossil fuel, when burned, they release a lot of carbon nanoparticles that we are inhaling. But if we see these nanoparticles, when it enters inside the body, it can create lot of diseases inside your body. So one of the most common disease that we are seeing nowadays, that is the asthma. And also we are getting lot of allergic reactions. But to study this nanotoxicity, we need to understand, before going for the human applications, we need to understand this toxicity on 3 different levels. So here my PhD student, and he is working in the area of nanotoxicology, where he has studied the effect of these engineered nanomaterials on in vitro and the in vivo labels. So as you can see here, I have made the 3 cartoons, where the first cartoon you can see here, this in vitro models that we just perform inside the lab, where we generally keep either the MTT or XTT assay to understand the cell viability or either the cellular toxicity. Sometimes to know how the cells are dying, we also go for the cell cycle arrest or the apoptosis assay, and even sometimes we perform the reactive oxygen species analysis. So whenever we are testing on the different healthy cells, then these cells we are using inside the lab, then these models are called the in vitro models. But before using for the human applications, any engineered nanoparticles, so as we are studying their toxicity, so we cannot only test on the in vitro models, we need to understand its behavior inside the in vivo models also. So there are 2 different types of in vivo models. One is the non-vertebrate model and second is the vertebrate model. So generally, a lot of scientists, they use the Drosophila melanogaster, like it is called the fruit fly, as a non-vertebrate model to study the effects of nanomaterials. And also, you can see like some of the researchers, they also use the zebrafish model. And also, when we go to the vertebrate model, then we need to use either the rodent models. And these rodent models either can be the smaller and the larger in size. So in vitro models and the in vivo models, these models are important to study the effect of nanomaterials toxicity. But when we go for the ex vivo models, we need to understand like why we are going towards the in ex vivo models. So you can see like you perform any experiments in the lab, and which provides lot of costs, and sometimes when you are replicating these results in the in vivo model systems, so these projects require huge money, and also it takes lot of time. So what scientists have done, they have just developed different types of ex vivo models to overcome the cost of these experiments, and also to reduce the time. So these ex vivo models are nothing, it is organoid models that we can develop from the healthy cells. And these healthy cells can be either from the brain, or it can be either from the liver and either kidney. And you can develop any type of healthy organoid from using these cells. So generally, you can say that this is called the 3-dimensional culture. So I think you know already about this, like there are 2-dimensional culture and the 3-dimensional culture. If you are testing the any type of drug or any materials on the cellular level and inside the lab on the plates, then you just using the cells and it is adhering on the surface or either it is floating, then it is called the 2-dimensional culture. But when you see these cells in the form of a spheroid or either in the form of an organoid, then it is called the 3-dimensional culture, and that models are called ex vivo models. And these models are developed. Why? Because it can reduce the cost of your experiments that you are spending for the in vivo models, and also it can reduce the time. So if you are using the healthy cells, then you can develop the organoid models, and if you are using the cancerous cells, then you can develop the cancer organoids, or you can say the tumor spheroid model. So here I have listed some of the experiments that we perform on the in vitro labels. So as you know, if you want to check the membrane integrity, like if you are testing any nanoparticles and it has disturbed the cellular membrane, so if you want to study the integrity, you need to perform the lactate dehydrogenase assay. And if these nanoparticles are just killing your cells, and it is showing the apoptosis, so you can study their morphology also with the help of different type of light microscopy, and also the fluorescence and the confocal microscopy. And the apoptosis study and the cellular necrosis you can study with the help of flow cytometry instrument, where you utilize different type of dyes like annexin of Fitzy, and these dye you need to understand which cells are apoptotic cells and which cells are healthy and the dead cells. And if you want to test the cellular viability and you want to see the cell death, then you use generally the Trappin blue dye and also the MTT dye, and you perform the live and dead assay. And if you want to see the damage of the DNA, then we perform the Comet assay, where we can see there is a tail which is forming from the which is forming when we run the gel, and where we are just running the DNA that is isolated from the treated cells, and you can see if it is forming the tail, then the DNA damage is happening for. And if you want to study the gene expression, so as you can see, like as Dr. Gopal has discussed about the qualitative PCR, so you can see the upregulation and downregulation of any genes using this qPCR machine. And as I discussed you, like when we are taking these nanomaterials for the intravenous applications, and if you are seeing these nanomaterials are interacting with the RBCs, and if it is damaging the RBCs, then hemoglobin will release. And that experiment is called the hemolysis assay. So you can use the ultraviolet spectroscopy and 570 nanometer, you can calculate how much hemoglobin is released in the outside the cell, and using the spectrophotometer, and you can calculate the hemolysis percentage. And if you are testing any inflammatory markers, it means these markers are nothing, these are proteins, or you are studying any immune responses. Either it can be the adaptive or the adaptive immune response, or either you can see the innate immune response. You can use either the Western blotting experiment, or you can use the ELISA to study these inflammatory markers. And when you are seeing like these nanomaterials has disrupted the ion channels, just like you know about the sodium potassium channels or the calcium channels, you can use the patch clamp experiment to see either these ion channels are working properly or not. And also, if you want to see the mitochondria has been damaged or not, so we generally perform the flow cytometry, and even you can use the microscopy also to see the like mitochondria has been damaged or not. And when you are studying the protein structure, so we use the circular dichrome spectroscopy, also the FTIR and the differential scanning calorimetry, and even the electron microscope, so that we can study the protein structure. So if the nanomaterials has damaged the protein structure, you can easily understand with the help of different types of spectroscopic methods. And as I told you, if you want to see the Ross generation, you need to use the DCFHDA dye, and this dye by which you can easily calculate the percentage of Ross inside the cells which is affected with the nanoparticles. And also you can see this Ross level with the help of microscopy also. So there are different types of assessment tools by which you can study, you can see the toxic effects, or you can study different types of toxicity tests. But these experiments we just perform on the cellular level inside the lab. So you can see here, I'm just giving you, for example, that we have conducted for our engineered nanomaterial. So you already know that we need to keep the experiment in the 96 well plate, where we have just seeded the healthy cells. And these healthy cells, we are treated with the different concentration of engineered nanomaterials. And we have incubated these treated cells for 24 hours, 48 hours, and 72 hours. And after some time, Then after each time point, like after 24 hour, 48 hour, 72 hour, after these 3 time points, we have added the MTT dye, and this dye is just used for to determining which cells are healthy and which cells are dead, like which cells are alive and which cells are dead. So we can easily calculate the percentage of viable cells and the non-viable cells. So you can see here, like we have tested the nanomaterials on healthy cells, and these cells we have taken from the repository, which is mouse fibroblast cells, and we seeded these cells and we tested the engineered nanomaterials at different time points. And you can see here we are getting the toxicity only 10 to 20%, and in the most cases the viability is more than 85% or the 90%. So according to the clinical standards, if we are getting the toxicity is less than 15% and or the 10%, and the viability is more than 85 to 90%, then these materials are not toxic to the for the in vivo applications, or we can say for the human use. But before performing these materials, or before testing these materials for the in vivo applications or inside the human, as I told you, we need to study their hemocompatibility also. So here I listed only 2 most important experiments. One is the biocompatibility, where you need to test the engineered nanomaterials on the healthy cells, and second is the hemocompatibility, where you need to test the engineered nanomaterials on the blood cells. So as generally you can see here in this figure, I have taken the RBC suspension, and these RBC suspension I have incubated with the different concentration of engineered nanomaterials. So you can see here, like after centrifugation, you can see on the bottom there is a pellet, which is nothing, it is just the RBC cells. While on the positive control you are seeing the red color, while in the negative control you are seeing the colorless solution. So here in the positive control we have used the distilled water due to we can say the isotonic solution. This distilled water entered inside the RBC, and due to high pressure, these RBC cells bursted and it releases the hemoglobin. While in the negative control and the other concentrations, we have used the PBS, and as this is the isotonic solution, that's why we have not seen any type of toxicity. So whenever we are keeping the hemocompatibility experiment, we always require distilled water for the positive control, and while the negative control, we generally take the phosphate buffered saline, or we can say the saline solution. So the same results I have replicated in the form of a histogram, where you can see we are seeing the toxicity which is less than 4%, like hemolysis on 20 mg per ml concentration of engineered nanomaterials. So as it is defined by the clinical standards that if you are taking any material and which is showing the toxicity less than 5% at any concentration, then you can use these concentration for the intravenous application. But if it is showing the toxicity more than 5%, then that concentration you cannot use for the intravenous application. So whenever we are testing the nanomaterials, we need to study the biocompatibility and the hemocompatibility of these nanomaterials on the healthy, also the blood cells. So after that, you can see, like as I have discussed the nanotoxicity on different in vitro models, similarly, you study the nanomaterial toxicity on different in vivo models. And these in vivo models, as I discussed earlier, it can be either a non-vertebrate model or it can be either vertebrate model. So I will discuss with you about the non-vertebrate model, but before going to that, I need to discuss about the vertebrate model, for example, the rodent model, like what are the parameters we need to. So you can see here, we need to study like the biochemistry, like whenever we are testing the nanomaterials inside the rodent model. So there are different types of toxicity that we check, and you already know about that, like the acute toxicity and the chronic toxicity. And this acute, before that we study the subacute toxicity, and also before studying the chronic toxicity we need to understand the subchronic toxicity. So there are 4 different of toxicity that we check inside the rodent model, that is the subacute, acute, subchronic, and chronic. Similarly, like these toxicity, we also study the systemic toxicity inside the rodent model. So there are total 5 toxicity we need to check inside the rodent model, or we can say in the vertebrate model. And whenever you are taking any engineered nanomaterial and you are just injecting inside the vertebrate model, either through the intravenous route or either through the skin route, whenever it is entering inside the rodent model, it will reach to the all the organ levels. Either it will reach to the brain, liver, kidney, spleen. So at different levels it will reach, and also it will go inside the blood, and where it will interact with the different cellular proteins, or either we can say it will interact with different serum proteins. So whenever once we test these nanomaterials inside the rodent models, and you have tested for different toxicity levels, and once the experiment finishes, you need to harvest these models, and you need to harvest their organs, and these organs we need to fix it, we need to make it slice, and we have to understand the histopathology. So you can see here in the histopathology, we make different types of tissue sections of different organs, and we use the hematoxylin and eosin dye, and we see either these nanomaterials has produced any type of apoptosis or necrosis, or even you can see the inflammation inside the tissue. And sometimes if you are not performing the histopathology sections, then most of the times if you use the antibody, then you can perform the immunohistochemistry. Similarly, if you have injected either by the skin or the intravenous route, then you need to calculate the hemoglobin, you need to calculate the total protein which is present inside the blood, the RBC level or the leukocyte count. And similarly, when we are going for the biochemistry level, then you need to do the alkaline phosphatase, lactate dehydrogenase, and different types of proteins that you need to study, which is present in the serum of the rodent models, which is treated with the different engineered nanomaterials. You need to calculate their levels so that you can easily know how much protein levels has been reduced or increased before and after injecting the engineered nanomaterials inside the rodent models. And whenever you already know that whenever we take any foreign protein inside our body, so lot of cytokines releases, and these cytokines also we can study, and these cytokines present in the serum. So there are different types of biochemistry procedures, hematological procedures, and histopathology that we need to do if you have tested in the rodent models. And sometimes as we use the rodent models also for the pharmacokinetics and thermodynamics study, so you collect the blood and you can study the toxic byproducts with the help of ICP-MS, and even we can perform the computed tomography or the positron emission tomography or the MRI. And if you are just conjugated your nanomaterials with any fluorescent dye, so you can see the fluorescence also, how these nanomaterials are entering inside the rodent models, where it is going, how it is degrading, and how it is eliminating. So how it is releasing from the body. So you can see the biodistribution, their clearance, and their elimination, all under the pharmacokinetics. So like that pharmacokinetics, we also perform the skin test, and sometimes you can see we also study the survival studies. And once these all studies we perform and we get the proper results, then we can go for the different clinical trials on the humans, where you can see the safety and the toxicity on human subjects for these nanomaterials. So these tests which looks like what we can say, like whenever we are testing these engineered nanomaterials on these models, as I told earlier, it requires lot of cost, and also other than cost, it requires lot of time. So that's why we have gone towards the development of lot of three-dimensional culture, so that we can reduce the cost and even the time to testing these engineered nanomaterials. So for just an example, I am giving you how we have tested these nanomaterials on the mice model. So you can see here we have developed a polymeric nanomaterial, where we have used the 25, 50, and 100 mg per kg of body weight of these nanomaterials to test inside the rodent models. And we have performed the systemic toxicity and the acute toxicity. We have injected at 0 day in 12 mice models, and the 5th day we calculated the body weight of these mice models, and also the 10th day. Similarly, we after the 10th day or the 11th day, we have just euthanized all these mice, and then we harvested different organs, the blood, so that we can go for the hematological and serum biochemical analysis, and also we can perform the histopathological analysis. So you can see, like here in the figure, if we check the on day 1, day 5, and day 10, and if you check the body weight of each mice, so you can see it is increasing in the trends, and which indicates that if the body weight is increasing of the mice, then these engineered nanomaterials that we have fabricated in our lab, it is non-toxic to the rodent models. And if the body weight is decreasing, then it means, it shows that these engineered materials are showing the systemic toxicity. So this is one of the tests that we have performed inside the lab on the rodent models. Similarly, as I discussed, either you perform the acute toxicity or the chronic toxicity, or either they are sub-toxic levels. You need to harvest the organs at the end of the day to study the apoptosis level, to study the necrosis or the inflammation. So you can see here, I have harvested the brain, heart, liver, lung, kidney, and spleen, and I have checked the apoptotic cells, also the necrotic cells on the inflammatory cells. So in the case of lung, we have seen only the small inflammatory cells, or we can say the inflammation at a small level. Or other than that, we found that with the help of pathological experts, that we could not get any type of apoptotic or necrotic levels in any organs, and also we could not see any inflammation inside any organs once we tested these once we tested our engineered nanomaterials. And the same experiment, as I told you, when we harvest the organs, we also take blood also from the each rodent models, and then we isolate the serum, and from that serum you can see we have isolated different proteins, and we have studied their labels. So as we provide the blood to the doctors, so that we can get the CBC count and all the protein labels, we also get the cholesterol levels, lipid profile level, and even you can see the protein level and those proteins which are required for your vital organs when we go for the blood screening. Similarly, the same tests we perform for the rodent models also, and we have tested the cholesterol, triglyceride, high density lipoprotein, low density lipoprotein, glucose, urea, and we have tested the monocyte, eosinophils, basophils, bilirubin, creatinine, similarly alkaline phosphatase, and you can see the C-reactive proteins. So there are different types of tests to be performed on these bloods that we have isolated from the rodent models, which is treated with the different engineered nanomaterials. So whenever we get the data, we need to just match with the normal range. If our data is not coming under the normal range, then we can see the parameters are affecting. Once our data is coming under the normal range, then we found that these engineered nanomaterials having no toxicity on any blood cells or any serum protein labels, and it can be used easily for any drug carrier purpose, or we can say for the tissue engineering purpose. So in this way, we study the toxic effects of nanomaterials on the in vivo models. As I discussed earlier about the vertebrate model, we have also studied on the non-vertebrate models. So on non-vertebrate models, you can see we have taken the fruit fly, which is Drosophila melanogaster, that we can easily handle in the laboratory. And one of my PhD student studied the polyester nanocomposite, which is an engineered nanomaterial, and we have tested on fruit fly, and we have gone for the different tests, such like a neural test or the genotoxicity test, and also we have done the cytotoxicity test, morphological test, and biochemical test. So whatever the tests I have listed here, all these tests we have performed inside the fruit fly. But here I have not given all the test details, only the few details I have given. So you can see one of the most important test is crawling assay, where you can see we have used the third install larva of the Drosophila melanogaster, and these larvae provide us the crawling behavior. If the larva is not crawling properly, it means it has defect in their neurons, and due to which these larva is unable to crawl properly and it cannot reach to the periphery. So it is a simple test so that we can understand the rhythmic behavior of the larva, and also we can understand like larva can move in a coordinated way or not. So it provides us the neurons behavior, and if the neurons has been if what the material we have tested, and if it is affected our neurons, then it means it has produced the neurotoxicity inside the larva. So what results you can get that the larva is not crawling properly and it will not reach to the periphery, and you also will see from the center to the some distance the larva will show more zigzag pattern. So it means the neuron has defect. But here we can see in the results with the control, larva is showing at all the concentrations the rhythmic behavior, and it is not showing any non-coordination. So you can see all the larva has reached to the periphery and it is showing the movement in a coordinated way, which indicates that when we tested these engineered polyester nanocomposite material inside the fruit fly or on fruit fly larva, so we found that it is not affecting any crawling patterns, which indicates that these nanomaterial does not show any neuronal toxicity. Similarly, we have tested these nanomaterials for the survival of the fruit fly models, and you can see these survival assay determines how many flies have been survived for different days, and we have tested the different concentration of engineered materials or this composite, and you can see like the survival patterns is decreasing when the time is increasing. So if you see the percentage of survival which is decreasing with the time, so you can see, moreover, it is just similar to the control, and you are not seeing the more changes at different concentration. It means that these engineered nanomaterials has no more toxic effects inside the like on the fruit flies, and due to which their survival rate has not been affected. So generally, it survives for these fruit flies are surviving for the normal days, whatever their lifespan. Similarly, we have tested these nanomaterials to study their phenotypic pattern. So we have taken the eyes, wings, abdomen, and the bristles, and you can see we have not seen any type of changes in the morphological features. If we take either the eyes or the wings or the abdomen or bristles, you see all the consent like at all different concentration, the treated fruit flies is showing the same pattern as you can see in the control, and which and also if you see the wing venation patterns, the wing venation patterns is also the same, and also on the thorax the number of bristles are same that we calculated, and also the abdomen, if we see, all the abdomen looks the same size. So it indicates that this nanocomposite have not shown any phenotypic alternation, and due to which these nanomaterials has no significant toxic effects on the morphological features, and also it has not shown any neuronal toxicity, and the fruit flies has been survived for their normal lifespan period. So these results we have published in American Chemical Society magazines, and also the journal has been considered our work to publish as a front page on the magazine. So you can see here in 2023 we have published this work on the front page also of the journals. So as I discussed, like we have used the in vivo models where we have taken the vertebrate or non-vertebrate models. But as I told you earlier, to do the experiment on non-vertebrate model is easy, and also it requires lesser money, and you can save the time. But whenever you are performing on the non-vertebrate model, you need to perform the test also in the vertebrate model. Then only your results will be confirmed. But these tests having high cost. Also, you cannot conduct easily within the time. So that's why nowadays my lab is also studying the effect of these engineered nanomaterials on organoids, where we are developing the three-dimensional culture of these cells that we have isolated from the different organs. So these cells actually we can take from the National Center for Cell Science, NCCS Pune, where they have the tissue bank and also they have the cell culture bank. So from where we have bought the cells and we have developed different types of organoid models. So you can see here the lungs, brain, intestine, kidney, and the liver. You can isolate the tissue and then you can separate the cells, and these cells you can utilize to develop the organoid models. And organoids are nothing, they are the three-dimensional culture of the cells. So what are the challenges we see in the organoid technology? The most important challenge is vascularization. Till now, we have not achieved the vascularization in our organoid models. And for getting the new vascularization, it is very difficult because we need to provide the growth factors so that the cells can develop the blood vessels and it can nourish each other, so that the cells could get us like cell can work sufficiently. And when it is developing in the organoids, so organoid is nothing, it is an organ, and this organ, if it wants to live for the longer time, then we need the vascularization also. But to get the vascularization is very difficult. So we are trying hard to develop the models, organoid models, where we can develop the blood vessels, and so that it can be vascularized. Also, acto-meso-endoderm interaction. So as we know, during the developmental pathway, like three germ layer forms, and these three germ layers interact with each other, and each germ layer requires to develop different systems like integumentary system, circulatory system, and respiratory system. So whenever we use the organoids, when we develop the organoids, we need to also study how the ectoderm, mesoderm, and endoderm is forming, and when the cells are interacting in these three germ layers, how they can lead to the development of organoid, which can be functional in later. And also, the most important challenge that we face during the organoid technology, like these organoids mature very fast. And if we are keeping these organoids for 10 days experiment, it undergoes cellular growth and also the division quickly, so that it reaches to the maturation, and then it undergoes the apoptosis, and then the size of the organoid reduces. So we need to perform the experiment before the maturation of the organoids. And the most important perspective is that these organoids we can easily use for the tissue engineering applications. But inside our body, there are some cells, they cannot be cultured easily. We need other cells also for the culturing. That's why we also sometimes require the co-culturing for those cells which requires to develop an organoid. So in our organs, there are some cells. If you take the pancreas, in pancreas, it is an exocrine gland. So endo and exocrine gland. So here we use two different types of cells to develop the pancreatic model. And the other important aspect is spatiotemporal control of signal. Because whenever you are developing these organoids, how the cells are receiving the signals, and so that it can develop into the fully organoids, and how they are interacting with the other cells. So as I have discussed the challenges and their perspective and what is required to develop these organoid models, but this technology is quite difficult but easy to do, but it requires lot of expertise. So these organoid models, scientists have used the nanomaterial toxicity on these organoid models, where they have developed the organoids from the different organs, and they have taken the cells from different organs, and then they have developed the organoids, and they have studied the different markers to see, like these markers, what are the changes comes in these markers, so that they can understand like what type of pathways it is affecting. So they have studied the apoptosis, the morphological change. So as I discussed in vitro models, the same experiments we also perform here, and also the gene expression analysis, like everything we need to do the similar. Only the difference is that in the in vitro model, we are taking the two-dimensional culture, but in the organoid model or the 3D culture system, we are taking the model in the form of organoid or either in the form of sporoid. But when we discuss about the experimental tests or the analysis, it is quite similar. So as you can see, like I have discussed about the ex vivo models, we need to also study the toxicity of these nanomaterials inside the human body. So when these nanomaterials goes inside the human, then it is absorbed by either the lungs or the intestine or in the skin, or it goes inside the blood, where it will distribute to the other organs. So it will go through to different organs, and where it will interact with the phagocytes, it will interact with the different organs or in their cells. And if it is reaching near to the phagocyte, then phagocyte, what they will do, they will just eat these cells, eat these nanomaterials, so that their effect will lose. And also once these nanomaterials will distribute, then as I said, it will start degrading, and during the degradation it releases lot of free radicals, which is responsible for the generation of reactive oxygen species, and also it can denature different enzymes and hydrolyze the proteins. So you can see their effect on the organs, and in the later stage, it will release these toxic products either through the mucus or the urine or the bile or the exfoliation or through the breast milk. So these are the four important parameters like absorption, distribution, degradation, and the elimination. That all these four parameters that we studied during the pharmacokinetics study. But whenever we are testing these nanomaterials inside the rodent models or inside the human, and we need to isolate the sample and we need to just isolate the protein, and these proteins we need to study using the NMR spectroscopy, either the HPLC or the mass spectrometry, so that you can see what are the toxic effects of these nanomaterials and how you can see when they are degrading, what are the degraded products they are releasing, that you can study by these spectroscopic methods. Also, how much toxic products is released and what are the toxic effects on different organs. So everything you can study using these instruments. And in the last, as I discussed about the toxicokinetic study, the last, you can see there are different types of computational approaches has been developed to study the toxicity of the engineered nanomaterials. So why we have developed to reduce the cost and also to replicate the data. So you can see whatever the data you are generating using the in vitro models, in vivo models, or the clinical studies, the same data you can see either it is replicating in the in silico models or not. And if you have produced the data from the in silico models, you can validate these data using different experimental methodologies. So nanomaterials, whenever you are designing, you can understand their design, and also you can predict the toxicity using the in silico models. And then if you are getting that these nanomaterials are non-toxic, then you can go for the different experimental methodologies inside the different, or we can say on different models, in vitro and the in vivo models. So you can study the size, shape, agglomeration, aggregation, and the chemical composition, solubility of these nanomaterials like we can see, like we can study the different physicochemical parameters of these nanomaterials inside the different models. And before testing these nanomaterials on the models, we can study like we can predict their toxicity by a different in silico models. So as I told you, like different types of experiments we perform on experimental level, the same results we can also produce with the in silico models. So whenever you develop any engineered nanomaterials, if you study the toxicity of these nanomaterials using the in silico models, you can actually predict what results you can get through the experiments also. So you can see here, there are different computational approaches for toxicity prediction. So I think some of you have been used the docking studies, read across study, and QSAR studies, and the structural studies, and even the expert systems we use to understand the toxicity or for the prediction of toxicity of engineered nanomaterials. And when we use them, then why we are using these computational approaches? Because it reduces the cost. Also, you do not utilize any type of animals for your experiment. Also, whatever the gap you see, you can fill the gaps using these in silico models. Like you can predict the data and you can fill these toxicity gaps. Also, it is a cost and time effective. You can study the risk assessment and the management. You can make the regulatory decision making so that these nanomaterials can pass the FDA guidelines, US FDA guidelines, or the CDSO guidelines or the ISO guidelines. And then it can be utilized for the human chemical applications. And also some of the mixtures you have prepared where you have used the different mixtures, you can also predict their toxicity. So these are the models like in silico models, also the in vitro models, ex vivo models, in vivo models. All these models are required to study the toxicity of nanomaterials. So as you can see here, I have given the list of toxicity that we can see inside the human beings. Like you can see the effect of nanomaterials on the damaging the eye, skin sensitization, skin irritation, damage, dermal penetration. You can see the allergic response, toxicity, gastrointestinal irritation or damage, neurotoxicity, immunotoxicity, endocrine disruptor. All these you can study on different in vitro models, ex vivo models, in vivo models, and in silico models. And if you are using the in silico models, you can predict the toxicity of engineered nanomaterials before testing to the different in vitro, in vivo, or ex vivo models. And once you predict, you can test them. And if you are performing the three-dimensional culture models, then you can reduce the cost, and so that you cannot test these materials on in vivo models. So here in our laboratory, we test the toxicity on in vitro models, ex vivo models, and in vivo models. Also, we just validate our data with the in silico models also. So I am very thankful to my all the collaborators and the funders from the DST SAR and the Indian Academy of Science. Also, I thank to my all the collaborators from the University of Edinburgh, Aalto University, NIT Rourkela, and IIT Madras. And I also thank to my all the students for working hard for to study the toxicity of different engineered nanomaterials at different levels. And I, in last, I thank to the Sharda University for providing all the facilities to perform such experiments and to perform to conduct such studies at university level. Thank you very much. Hello.
Thank you, Dr. Piyush.
Yeah. I'm sorry, sir. It is quite it is quite long talk.
Any questions from the audience sides?
I think I have gone beyond the time.
Yeah, hello, sir. Sir, any questions from these audience sides? So one question from my sides.
Yeah, sir.
So what are the main factors contributing to the potential toxicity of nanomaterials in biomedical applications?
Sir, the most important, as I discussed in the previous slide, first is the precursor. What precursor I am using to develop the nanomaterials. And before considering the precursor, I need to understand what application I am going to perform. So in my lab, we generally perform the drug delivery experiments and the tissue engineering experiments. And for the drug delivery experiments, we try to develop the negatively charged nanoparticles. And we consider our route intravenous. So if we see the RBC has negative charge. And if we are preparing the negatively charged nanoparticles, then it will not interact with the RBCs, so that we cannot see the hemolysis. And that's why we can easily use for the drug delivery, applications. Second, as I discussed about the precursor, so precursor determines the charge. So what is the net charge on the surface, which is also an important factor. So as I told you that RBC has negative charge and we are developing the negative charge nanoparticle, then it will repel. So we cannot see the hemolysis. So these are the two important parameters that we take for the developing the nanoparticles. But we cannot eliminate other parameters, because size is also an important concern. And also, other than size, the shape is also an important concern.
Yes.
So actually, we do not eliminate any parameters. Whenever we develop the nanoparticles, we try to take all the parameters for our consideration, so that we can develop a smaller size nanoparticle, and it cannot show any type of aggregation, and we try to develop the negatively charged. We take the precursors which are naturally obtained, so that we cannot see any toxic products after degradation. And also we try to develop a rod shaped and the disc shaped and even the spear shaped nanoparticles, but it completely depends upon application.
Okay.
Yes, sir.
Thank you, sir. So next question is my side. So what are the challenges associated with the visualization of invention of organoids?
Sir, challenge is that, sir, as I discussed just now, that vascularization. And this vascularization we are trying from the last 10 years. And the thing is that when we are developing any organoid models, and we want to develop into a fully organs. So whenever we are taking any growth factors, and these growth factors we are delivering, it is not reaching to the cells properly. And for minimizing that, what we have done, we have taken the nanoparticles so that they can deliver these growth factors to the cells. But the thing is that we cannot, whenever we are taking any growth factor, just like you take like VEGF. So when we are just uploading these growth factor on the surface of nanoparticles, after some time we see that growth factor has been denatured.
Yeah.
And we do not know, like we have tried with the development of hydrogels also, and we have tried to deliver this growth factors with the hydrogel.
Yes.
But the thing is that if we are using the hydrogel, then we cannot take for the longer time. It is degrading fastly. So vascularization is a challenging issue that we are still facing. And we in my lab, we have also performed the decellularization. I think you know, sir. So
Yes.
we have taken the organ and we have decellularized the organ, and then we try to recellularize the organ. So recellularization also, we are not able to get the vascularization.
Okay.
And
Thank you.
the same thing we also tried. The nanoparticles, where we have loaded the protein factors on the surface of nanoparticles. But I do not know what is happening when it enters inside the cells. These growth factors denature.
But sir, when we will do for these in vitro models, how will contribute to the assessment of nanotoxicity?
Sir, in vitro models, I do not rely and I do not believe on their results also, because if you take the interactions and the environment, what you see environment inside the body, which is completely different that you see in the in vitro model system. So generally in vitro model systems, if we are taking, sir, environment in an important consideration, the cells is also stressed and they have been growing from the longer time with different conditions, and they have been sensitized and they have experienced all that stress. So they are surviving. And if you isolate any cells just from the organs in 1 or 2 days before, and you grow in the lab, and that cells will not grow because they have not seen such stress. That's why on in vitro systems, when we test, we do not rely their results. We try to replicate these results in the in vivo model system. If we have money, if we don't have money, then we go for the ex vivo model systems.
Yes, sir.
Yes, sir.
Thank you, sir.
Yes, sir.
Any questions from the audience sides? Or else we will conclude to the sessions. Sir, any questions?
Yes, sir. How are these nanoparticles? Yeah. Yes, Samriddhi.
Sir, I wanted to know that you're telling us about the experiments which had been conducted on the invertebrates and the vertebrates, in which there was no significant change was seen after the nanoparticles were, injected into them, or they were put into the organism's body.
Yes.
But at the same time, we are studying about carcinogenicity and all those ill effects which nanotoxicity can have. So, sir, my major question is in which situations does this toxicity arise? Because in experimental things, only what we saw from the slides was that it was the lung tissue which showed major inflammation, right? Other
Yes, yes,
tissues, they remained almost same. So,
yes, yes.
how would we know that when are we prone to nanotoxicity?
Yeah, so that is a very good question, Samriddhi. Actually, what results I have shown you, that is acute toxicity, and
Okay,
which we have conducted only for 10 days. And
all right.
actually, this experiment we should conduct for more than 30 days, or we can say for the 60 days, because you see the effect of nanoparticles when it undergoes the chronic effects more.
Okay.
In
Hmm.
acute effect, as you know, if you are staying in Delhi, you are affecting every day with the carbon nanoparticles, because there is a lot of pollution in air
Right.
pollution in Delhi.
Yeah. Hmm.
But initially what you will see, you will get the allergic reactions, and you will see that some inflammation. But when it will go for the chronic stage, then only you will get that your lung is affected and the lung cancer has been arised. And also you have got different types of respiratory disorders. But these effects comes in the chronic stage. So if I perform these experiments for the chronic stage, then surely I see the necrosis and apoptosis also inside these organs.
Okay, so sir, you're telling me that it takes like a bit more time for
More time.
biomagnification to actually be seen and observed.
Yes, we need to give more time to understand the behavior of nanoparticles. And it
So then
depends upon,
can we classify this as a slow poison then? Because it is slowly.
yeah, it is a slow poison only. Initially it gives you only the small inflammation, and slowly it goes to the allergic reaction, then it goes to the asthma, and then it will lead to the respiratory tract infections. And once it will not be treated on time, then it can generate the lung cancer also.
So sir, is it completely asymptomatic or are there, minor
No,
symptoms or signals which we can detect? Because
sneezing.
like in cancer also, at the second or third stage, generally it is seen and then it's too late to, save the patient.
Yeah,
So are
you
there
see
any?
the you
Hmm.
see the signal from the day one, because once you inhale any type of nanoparticles which is not meant for your body, you
Yes,
start
sir.
getting the sneezing. You will
So
get you will
allergic reactions basically.
allergic reactions. So you
Okay.
get you get these signals from day one only. But
Oh, all right.
I'm telling, once it goes to the longer stage, like a chronic stage, then you get the severe toxic effects. And
All right.
lung cancer, why I am telling, it is
Hmm.
not arise earlier. It will take time to generate the malignant cells in the lungs after a long exposure.
All right. Okay. Okay,
Yeah.
sir. Thank you, sir.
Okay. Yeah. So Sumit is asking, as metallic nanoformulation are toxic in nature, then why we prepare? No, no, no. Each metallic materials is not toxic. As you bear the gold or the silver, these are inert metals. They are not toxic in nature. And also, when we develop the metallic nanoformulations, we need to understand how these materials are designed, because these materials, when we discuss in metals, these metals how they are forming, like we study on the atomic level how these metals are arranging with each other. If their arrangement is not in a proper manner, then it might be a toxic. When the metals are in proper arrangement, then it cannot be toxic. And also, like we discuss about the zinc oxide, and we discuss about the iron based materials. These materials, if they degrade also, then it cannot be toxic to the human body, because these metals are acting as a cofactor for different enzymes in our body, such like iron we utilize to develop the like a hemoglobin. Also magnesium, that we utilize for the DNA replication. Zinc for the carbonic and hydrogen chain. So whenever we are selecting any material, we always understand which material or which metal we are selecting. And one question is from Abhay. Sir, I would like to ask how are these nanoparticles are used to deliver the insert drug or recombinant DNA in the target cells? Yeah, so generally, Abhay, we utilize the lipid-based nanoparticles to deliver the recombinant DNA molecules, because as you know about the COVID vaccine, where we have used the recombinant DNA. So in the COVID vaccine, that recombinant DNA we have encapsulated inside the lipid nanoparticles, and when we injected it on the skin, these nanoparticles fused, fused with our cellular membrane, and it delivered the drug inside, and it delivered the recombinant DNA inside our cell. Then our cell used these recombinant DNA and integrated into our genome, and then it start producing different DNA products, and also it undergoes the transcription, and then it forms the after the translation it forms the protein. So generally, if we want to see the delivery of recombinant DNA, then we use the lipid-based nanoparticles, and the drug you can deliver either with the lipid-based nanoparticles or polymer-based nanoparticles. But whatever the nanoparticles we are developing, we need to see what are the degraded products it is releasing, and either these products are toxic or non-toxic. So that's why when in my lab we develop any type of nanomaterial, or we can say the biomaterial, we study their toxic effects, we study their degradation, and what are the products they are releasing. And once we found it, like all the products that has been released after the degradation, which is non-toxic, then only we utilize those biomaterials for the drug delivery and tissue engineering application.